2020-2021 Lineage B 1.1.7. Also known as 20I-501Y, V1. Variant of concern 20DEC01 more transmissible than wild-type SARS-CoV-2 and was detected in November 2020 from a sample taken in September. During the COVID-19 pandemic in the United Kingdom, it began to spread quickly by mid-December, and is correlated with a significant increase in SARS-CoV-2 infections in the country. This increase is thought to be at least partly because of one or more mutations in the virus's spike protein. The variant is also notable for having more mutations than normally seen. As of January 2021, more than half of all genomic sequencing of SARS-CoV-2 was carried out in the UK. This has given rise to questions as to the variant's origins, and how many other important variants may be circulating around the world. On the 2nd of February 2021, Public Health England reported that they had detected limited number of B1.1.7 VOC202012-01 genomes with E484K mutations, which they dubbed variant of concern 202,102-02. This mutation is also present in the South Africa and Brazil variants and may reduce vaccine effectiveness. The variant is known by several names. In British government and media reports it may be referred to as UK COVID-19 variant, UK coronavirus variant, the new variant or, particularly outside the UK, as the UK variant or British variant or English variant. It is sometimes called the Kent variant after Kent, where the variant was found. In scientific use, the variant had originally been named the first variant under investigation in December 2020 by Meera Chand and her colleagues in a report published by FE on 21 December 2020. In a report written on behalf of COVID-19 Genomics UK Consortium, Andrew Rambo and his co-authors, using the phylogenetic assignment of named global outbreak lineages tool, dubbed it Lineage B 1.1.7, while next strain dubbed the variant 20I-501Y, V1. The name VOC202102-02 refers to the variant with the E484K mutation. B 1.1.7 was first detected in early December 2020 by combining genome data with knowledge that the rates of infection in Kent were not falling despite national restrictions. The two earliest genomes that belong to the B 1.1.7 lineage were collected on 20 September 2020 in Kent and another on 21 September 2020 in Greater London. These sequences were submitted to the GISAID sequence database. Backwards tracing using genetic evidence suggests B 1.1.7 emerged in September 2020 and then circulated at very low levels in the population until mid-November. The increase in cases linked to the variant first became apparent in late November when Public Health England was investigating why infection rates in Kent were not falling despite national restrictions. FE then discovered a cluster linked to this variant spreading rapidly into London and Essex. Also important was the nature of the RT-PCR test used predominantly in the UK. Thermo Fisher Scientific StackPath COVID-19. The test matches RNA in 3 locations, and stopped working for the spike gene due to the HV69 to 70 deletion. A deletion of the amino acids histidine and valine in positions 69 and 70, respectively, of the spike protein, in B 1.1.7 spike protein. This made preliminary identification easier because it could be better suspected which cases were with B 1.1.7 through genome sequencing. Although the variant was first detected in Kent, it may never be known where it originated. Discovery in the UK may merely reflect that the UK does more sequencing than many other countries. It has been suggested that the variant may have originated in a chronically infected, immunocompromised person, giving the virus a long time to replicate and evolve. Mutations in SARS-CoV-2 are common. Over 4,000 mutations have been detected in its spike protein alone. According to the COVID-19 Genomics UK Consortium, VOC202012-01 is defined by 23 mutations, 14 non-synonymous mutations, 3 deletions, and 6 synonymous mutations. On 2 February 2021, Public Health England reported that they had detected Limited number of B1.1.7 VOC2O2012-01 genomes with E484K mutations, which is also present in the South Africa and Brazil variants. This mutation may reduce vaccine effectiveness. On the 9th of February 2021, it became known that some 76 cases with the E484K mutation had been detected, principally in Bristol, but with a genomically distinct group in Liverpool also carrying the mutation. A week later, a research and analysis report from FE gave a total of 77 confirmed and probable cases involving the E484K mutation across the UK. In two variants, VUI202102-01 and VOC202102-02. The latter described as B1.1.7 with E484K. On the 5th of March 2021, it was reported that a B1.1.7 lineage with the E484K mutation has been detected in 2 US patients. Researchers think that the Oregon variant has evolved independently. Estimates of VOC2021-01's transmissibility have varied greatly across different studies. In a preprint, the Center for the Mathematical Modeling of Infectious Dis.